Good evening, everybody, and welcome to our monthly meeting of the New York City Neighborhood Naturalist Group, which is part of the NYC EcoFlora project. My name is Lydia Paradiso, and I'm a doctoral student at the New York Botanical Garden and a graduate fellow with the New York City EcoFlora project. NYBG created the EcoFlora project back in 2017 with a grant from the Institute of Museum and Library Services. The EcoFlora program is a community science project with three main goals: to investigate urban ecosystems and urbanization, to support open source biodiversity data, and to increase understanding and appreciation of plant life. To date, nearly 30,000 observers have made over 820,000 observations of plants and their biotic relationships in New York City. You can explore the observations on the project page on a naturalist, which I'll link along with some other links in just a minute. Each month, we also sponsor an EcoQuest challenge, which is a sort of scavenger hunt to encourage New Yorkers to observe a particular species or a group of species and their ecological partners and post them on a naturalist. This month's EcoQuest is called Encounter Evening Primrose. Common evening primrose, the scientific name is Oenothera biennis. It's a disturbance-adapted wildflower that can grow up to 6 feet tall. Its yellow flowers, which open in the evening, are pollinated by insects such as hawk moths and the evening primrose sweat bee. Because these interactions occur in the evening, they're underobserved. So part of the goal for this EcoQuest is to observe evening primrose flowers in the evening or nighttime being visited or by pollinators or be opening. But of course, you can also observe them during the day, as I think that's probably when most people are out. So I'm gonna link here in the chat. We've got 3 links. The first one is our general EcoFlora page on the NYBG website. The second one is the overall iNaturalist project, and the third one is the EcoQuest. Any other announcements? We will have continue to have our kind of plant walks. So keep an eye out in your emails for updates on those, because we'll be all over. So far, we've been in the Bronx and we've been in Brooklyn. So looking forward to getting to the other boroughs as well. And so now I will turn it over to Tomi to introduce our speaker.
All right. So before I introduce our speaker, Gema, I'm going to begin with a land acknowledgement by recognizing the original people of this land in New York State and recognize it as the land of the Lenape. As well as recognizing their deep connection to the Lenape-Hoking homeland. As an organization that is dedicated to advocating for the plant world, we must acknowledge those who are original stewards of the land and believe in the importance of acknowledging the settler colonial genocide perpetrated against the community and the resilience of the Lenape, who still today continue to resist erasure. Okay, so to introduce our speaker, Gema. Gema is an assistant professor with the Department of Social and I just practiced how to say this, but Myrmecophilus insects at the Museum Institute of Zoology in Warsaw, Poland. She earned her undergraduate and graduate degrees from the University of Cordoba in Spain. Her PhD thesis focuses on the use of ant species as bioindicators to study the role of urban greeneries in the global strategies of conservation. In 2014, she moved to Poland to her current position. Deeply in love with ants, Gema's main interest of research focuses on urban ecology, using ants as a model species with the aim of getting a better understanding of the urban ecosystem's functioning and contributing to find a balance between development and biodiversity conservation. She also worked with her colleagues in Warsaw on a series of projects related to ant personality, vibroacoustics, and forest ecology to better understand a knowledge that greatly helps to understand the social structures of ant colonies and their relationship with the surrounding habitats. So that is our introduction for Gema, and then you will get to know her research better as she presents her presentation.
Okay, thank you very much. First, I would like to thank you for the invitation. It's a great honor for me to be speaking to you today and show you the results or some preliminary results of my current project. And also, I would like to apologize in the case that I'm saying some weird word or some weird pronunciation, because as you can see now, I'm not native English speaker. So, I will start sharing my screen. And please let me know if you can see it properly. Okay. So I don't really know how much you know about ants, but in my case, I started to know about ants when I was a student. My professor was is a myrmecologist at the University of Cordoba, and he was deeply in love with the ants, so he transmitted it to me. He was always also working with the urbanization, so my career started like that. Later, I started doing some other thing, learning about ants in other aspects, and but I still keep working with that. Because from my point of view, and let's say that despite I will show you today my results, I will talk you about my point of view, and it means that maybe some other person cannot agree or they have some other opinions. In that case, if you share it, it would be nice also. So I think that ants are very important, and urban ecology is even much more important nowadays because we are suffering a loss of biodiversity. One of the main factors for that is the habitat loss. And urbanization is a fact. We cannot fight by that. We cannot come back to the like 500 years ago or 1,000 years ago. We are used to some, let's say, comfortable life nowadays. We have our cars and we need the roads for that. We have our homes, we have electricity. So this comfortable life is something that we won't renounce, but also it's something that we cannot keep ignoring because it's affecting animals. So my point, I think, and maybe I'm a bit wrong, but I think that the best option could be to find a balance between our comfortable life and still trying to modify the structure of the cities using the urban gardens or these urban greeneries that offer a refuge for the biodiversity to find this balance. And this is the base of my studies and the base of this talk today, ants in the city, influence of urbanization of arthropods using ants as bioindicators too. Well, since I don't know how much we know about ants, because this is not like the lions or the monkeys, we all know about these beautiful animals. Ants are a bit hidden. We have them everywhere, but still for us, when we check them, they all look the same. But it's actually not like that. We have more than 15,000 species described in the world. And focusing in Europe, I know I'm talking for you in America, but my knowledge is not that deep about the American species. In Europe, we have more than 600 species, and I'm from Spain. In the Iberian Peninsula, we have around 300 species, and in Poland, where I'm working, we have 100 species, which is a lot. So even if they look the same, we have many ants. And all those ants, they have different functions in the ecosystem. They promote the soil aeration, they spread the seeds. They also are important of the food web because they are food, but they are also predators. They are cultivating fungus. They are doing different things at different levels of this food web. They are also a pest control because they are predating, so they are somehow reducing the number of these pest insects affecting a habitat type. And they also are hunting. Well, there are some studies telling that they are mostly focusing on hunting those animals that are in larger proportion, which is actually a pest. They also helping for the for the soil nutrients because of this food web. And they are pollinators. This is something that is not known for ants, because most of the ants, most of the pollinators we are talking about, or we are thinking about, are the bees and so on. But ants, they are not that important, of course, as pollinators, but they have a function as a pollinators. They are also, their bodies are covered by a small seta, and they are also collecting the pollen from the ants, from the plants when they are moving, and they are moving to other plants to collect the nectar. But also, there are even some plants that they have modified their structures to be pollinated by ants, or visited by ants and secondarily pollinated. So they are they really have a very important function in the ecosystem. But, oops, what happened here? But they also are very linked to the ecosystem because of that. So it means that if the ecosystem is having some changes, also the ant communities, they are suffering these changes. So when you have animals that are fully linked to the habitat type, and small changes in the habitat are strongly reflected in their communities, or in the their abundance, for example, their distribution, and so on, different aspects of their ecology or their structure, they are considered very good bioindicators. So taking that into account, let's think a bit and let's go back a few years ago when we were kids. For some of us, it's larger time than for others. So when I was a kid and I went out from my home, I had a small field. There were the road, because I'm from a small village. So there was the road, the houses, and then they had some meadows behind. If I'm thinking nowadays, all of this landscape has changed a lot. Those meadows are not anymore. And if I go out from my home, I have the road, houses, more roads, more houses. And this is somehow like affecting us when we want to go, when our kids want to go out for playing, or if you want to go out to do, to have a walk in the meadows, to have some contacts with nature. So coming back to the present and thinking about our own experience, this experience is stronger in the case of the animals, or in this case, arthropods, that they cannot move so freely as we can taking a plane. And this is affecting their communities. The problem is, like when we have seen here the role that they have in the ecosystem, if their communities are affected, also our habitat types, and it will have several serious consequences in all the aspects that we all know in the ecology, because unfortunately every day in the news we have several bad news about our ecology situation, ecological situation, climate change, the global warming, or all of these fires we have in different woodlands. Let's say a series of them. So one of these, as I was talking before, one of these factors, or of these changes, is the urbanization. So now we are going to think about the urbanization, all these changes that we have seen in our villages, cities, and the landscape in general that we are inhabiting, and we'll see how it is linked to our communities and how ants are showing that. So the starting point of urbanization is to think how this landscape changed, because we have built huge cities. And these huge cities are the focal point for work and business. Many people is moving there. So these cities have to grow and grow, and the surrounding habitats are decreasing. The natural habitats are disappearing. So this reduction is also the loss of the nesting habitat for many species, ants, birds, and so many like reptiles and so on. But if we think in the city, we also have our greeneries. We have botanical gardens, we have parks, and they can still have a function as a refuge for these species. So we will analyze how they are built, how the structure of these greeneries is important for our ant species or for the bioindicators, and how it is important as well for other animals. And also a different aspect that is not the structure of the city, a different aspect of urbanization or effect of urbanization that is not the structure of the city is the urban heat island effect. It means that if you are in the city center, the different structure of the city, the high buildings, the pavement, and the different compounds of the city is warming during the day. And during the night, it's keeping also this heat because the airflow is lower, and this pavement, the concrete and the buildings and so on are absorbing a lot of heat during the day and releasing it during the night. So there is not a refreshment of the city if you compare it with the natural habitats. And this is also changing, and this is really a change in their in these environmental features, like well, the temperature of the city or the temperature of the habitat, and it is also having an effect. So analyzing the environmental factors or the characteristics of characteristics of the city or the habitat types and how it is linked to our species, we will check the age of the habitat, how it is important, and the habitat structure, which is the microhabitat or the components of the habitat, and the character or nature of the habitat, if it is natural or managed. I mean, or in other words, if you have natural habitats or urban habitats. So to start checking how the age of the habitat and also the characteristics for the microhabitat we have, we started a study in the campus of Rabanales during my PhD. Here is a perfect site because we have this small garden, which is super young garden. It was built just a few years before my study. And then these other 3 gardens, exactly inside of the same campus, university campus, surroundings are the same. So surroundings habitats are not a different, or they do not influence in a different way the different gardens, and they are super old gardens. When I'm telling that, it's like they were when the study was carried out, these gardens were like 20 or between 20 and 30 years old, and this Aulario, the small garden here, it was only 3 years old. To carry out the study, we placed pitfall traps. What's that? This is small glass different people is using different sizes. And inside we are putting water and a drop of detergent. You place them at the ground and leave them for during 48 hours, 72, it depends on the habitat. For example, if you go to warmer habitat, the time placing that the pitfall trap will be working in the ground will be shorter than if you go to colder habitats. Once you place that, wait, ants will drop inside, and then start a game for the identification and the cleaning of the pitfalls. As a result, we saw that when you are analyzing the different gardens, you have the small garden, the young one, have a composition different to the 3 old gardens. And these 3 old gardens are quite similar among them. If we are comparing to the natural habitats at the surroundings, there's no colorful dots, let's say, you have exactly the same. All natural gardens, the old gardens are more similar to the natural habitats, but still the young garden show many differences. And how do you know that, or how do you see that? When you analyze the data, you can see that different other species are linked to the different gardens. So the young garden one is linked to some species that are very proper or very anthropophilic species, like, for example, Cardiocondyla mauritanica. It's not a native species, it was introduced and is very linked to the habitats or the human habitats. Or for example, you have Tapinoma nigerrimum, a very generalistic species also present in all habitat types. Or you have Tetramorium species, the pavement ants. However, if you go to the old gardens, you start having some different species like Temnothorax species that are more linked to the wooded areas or old habitats. Or for example, you are having Formica species that are typical species for gardens. So when you are analyzing how it's happening, how you have a young garden with a composition, and you have old gardens with a different composition, and you want to see how is the trend for these species very linked to the gardens, or the most important species showing us the differences between the gardens, you see here that you have, for example, Lasius grandis. This is the most or the common garden species in South Spain. When we started the study in the 2005, the young garden, this graph is all about the young garden, how it's developing along when it's aging. So the young garden is showing that Lasius grandis, the typical species from gardens, is colonizing this garden along the years. Same is happening with Formica funicularia, and same is happening with Solenopsis species. This is a species usually linked to major habitats and linked to other species. And for example, Mesobarbarus. This species is not typical from gardens. They do not like it. They are very big-headed species like your Veromesor, for example. So they are having higher number during the beginning of the study, and is decreasing to finally disappear at the end of the study, because the remnants of the original population in these habitat types is disappearing when the habitat or when the garden is getting major or is aging. So this is, we can see how when we transform the habitat, also the different populations, depending of the different species, depending of their ecology, is changing also their trend, either increasing their number, arriving and establishing their populations there, or disappearing if they are not from this habitat type. So why it is happening? Well, our species, our ants, as bioindicators, as good bioindicators, they are linked to some characteristics of the microhabitat. We have some species that they need the bare ground to nest, or as I said, the Mesobarbarus is not able to move freely among the grass because they are not so fast on the species, and this big head is not helping a lot. So when we were analyzing the habitat characteristics during 10 years, we saw that our different species inhabiting these gardens are strongly linked to the different characteristics of the different structures. When we had like a tree, or we had a shrub, when we were having bare ground, when we were having grass, rocks, for example, or the borderline between the grass and the sidewalk. This is strongly linked to the other species that we can find in a habitat, or what is translated in a different, in a more easy way. If we want a larger number of species, we need a larger number of components in the habitat. So when we are building these huge extensions of grass in our parks, it's a playground, but we are ignoring the fact that we need to put some stones, or we need to put some shrubs or some trees. What we are doing is limiting the presence of many other species, each one with a function in the ecosystem. And how we are using it, this is in the same pool. So when we are linking the different other species to the habitat characteristics, let's say what they need to be there and what we have in the habitat, and we compare it with what I should have in that place, I can analyze how many species I have, showing me the presence of disturbance, and how many species I have, showing me how mature and well preserved is this habitat type. And the proportion will show me that, for example, as I said before, the young garden one is not well preserved, but as we know, it was young, so there is the time needed to get this proper or the correct function, the real function in the ecosystem. However, when I'm analyzing one of the other major gardens, I'm checking the species I have, and I see how the maturity and the number of ants, indicators of maturity, is jumping from only a 10% in the young garden to a 38% in the major garden, which is a very, let's say, a very well proportioned and showing me a very well conservation in this habitat type. So, well, now I jumped. Oh, sorry. So, taking all of this into account and knowing that different habitat types, different structures, and different compositions will allow the presence or not of different other species. When I arrived to Poland, I wanted to put in practice all of these things that I was taking in South Spain and see how it is working in Poland, a place with a totally different habitat type and also a place with a different weather. So in Warsaw, I had the chance to analyze how, when you are putting together all the components of the habitat, and you have these different types of macro habitat or habitat type, how it is working in my ant community. And I had the chance because you had 2 botanical gardens within the same city. You have very large parks, the typical standing lawns of grass here. And we also have 2 forests or remnants of forest within the city. So the city was growing and they were trapped inside. So that's a very amazing situation to study the habitat type, how it is influencing my biodiversity, and without any other influence, because the influence of the surrounding will be exactly the same in all habitat types. So here is when I carry out the study of the 3 categories of urban green areas and the effect of the different management on the communities of ants, spiders, and harvestment. Of course, with the collaboration of all of these people, my colleagues, and one expert in spiders, because my knowledge about the spiders is quite limited. In this study, as I said, we wanted to check how the structure of these 3 groups of bioindicators are showing me the effectiveness of the habitat type of, as a refuge of the biodiversity. And also, we wanted to know what is it we need to improve the maintenance and conservation of the urban greeneries and their biodiversity. To do that, we sampled all of this habitat, all of this area. This and these are the 2 forests, and here we have the 2 botanical gardens. In total, we found 34 species of ants, which is 35% of the Polish myrmecofauna, 83 species of spiders, and 11 species of harvestmen, which is a quite low, high number, because we also use, again, the pitfall traps as a methodology for sampling. And if you are using one methodology, usually there are some cryptic species that are not reflected in your results. So that's a really nice, a really good number of species, taking into account that we are always working in this artificial environment. When we check the habitat type, we also see how all 3 habitat types are differing in their composition of ants. And botanical gardens are they are having like more ant species than the parks, and also is having like same number of species or not significantly different number of species when you compare it with the forest, with the woodlands or the urban woodlands. But not like parks are again showing a lower biodiversity than the woodlands. So it doesn't matter how you are comparing, parks are the less diverse habitat type. And also we found that in parks, we found the Lasius neglectus, one invasive species in Europe. And we also found Phaidole megacephala, but this invasive species shouldn't be taken into account because we only found 1 worker, and in further studies we never found the nest. So it seems that just it arrived with some ornamental plants. So when you are analyzing this biodiversity, if you go and check which are the proportion of generalist and specialist species, And this is very important because specialist species are strongly linked to the habitat type. So small change will make them disappear, and of course, they will not be able to fulfill their work in the habitat. And generally, species are more, let's say, plastic. They also have a role in the ecosystem, but they are not that specifically linked to the habitat type. So when we are checking how the different habitat types are affecting to the presence of a specialist species, we see that the lowest number of specialists is in public park, whereas the highest number is in the botanical garden. When we are jumping to the spiders, again, a bioindicator group, we find the same. We still find same big differences between urban parks, urban forests, and the botanical gardens. And again, parks are lower or are having a lower biodiversity than botanical gardens, as well in this case that woodlands. One more time, unfortunately, this is common in urban habitats. We found one invasive species, the Mermesus trilobatus. And analyzing again the two different groups, specialist and generalist species, again, the lowest number of specialist species is present in urban parks. So finally, checking the harvestment, it seems that the harvestment, they have a high mobility, so they are not as good bioindicators as I thought. And or as it told me, this is something that I learned with this study. And they are a bit more mixed everywhere. They are really anthropophilic species, so they are not showing real difference between the habitat types. And we also found the two invasive species here. So what we can see from this study is that with our changes in the habitat, we are changing the structure and the composition of the community, but also we are introducing species. And the spider ants are having a very important role, as I said, for example, as biological control for pest species. When we are not really focusing on the influence or the importance of our introductions, when we are introducing plants, when we are introducing other animals, we are somehow, when we are doing these introductions in a very sensitive habitat, like urban habitat, where our native species are not 100%, or they are not strong 100%, we are also risking to have, unfortunately, some negative aspects, as for example, a high abundance of invasive species in the villages near to the seaside, very susceptible to these interactions because of the weather. We are also changing our habitat characteristics, and we are affecting different communities, also plants. We are facilitating the introduction or the coexistence of invasive species. And despite they only look sort of invisible, they really can be aggressive and have a high effect. So to conclude, we can see that urban environments in general are much more rich than we commonly think, because usually when we are thinking about our urban environment, we think they are artificial, so they are not rich in species. But actually they are very rich. And it's up to us to build this beautiful place for these a suitable place for many species to increase or to enhance the roles as a refuge of biodiversity. And in order to do that, we have to think that the structure of the habitat has a very important role in the number of species. So when we are thinking about our garden, or for example, when we have to design the parks that they will be in this neighborhood, we have to think that maybe with if we are leaving some corners and then in a more natural way, if our selection of plants, of these ornamental plants, are more linked or are more close to the selection of native plants or local plants instead of these exotic ones, because in all countries we have very beautiful and very large number of native plants. Or when we are creating or we are constructing this mosaic of microhabitats, we can increase the number of species, and we can compensate this habitat loss with urbanization, with this refuge for biodiversity. And we have to be extremely careful with the introduction of these ornamental plants, or even some things, some other materials from constructions, because they are the main mean of transport of many invasive species. This is something that, for example, in botanical gardens is very controlled. They have very, well, you know much better than me. Plants have to follow a series of check-ins and no soil and so on in order to avoid the introduction of these invasive species of arthropods. And, sorry, it came here. As we see, ants are very useful bioindicators too. But for example, there is a big problem when we are using these ants. And it's like usually we have larger lists with the different species and their bioindicators. So for example, if I have Myrmica loba, they are bioindicators of, for example, like maturity or perturbation, depending of the place I am. But usually this categorization is made in base of natural habitats, how they are or where they are in natural habitats. But we cannot think in that way when we are analyzing urban habitats. And I think this is something a bit that is not really taken into account yet. That if we are analyzing these artificial habitats, we have to change all of this categorization. And despite we have some species considered indicator of perturbation in some natural habitat types, maybe in this small area where I'm trying to concentrate as much number of local and native species as possible, maybe the presence of this species is totally opposite. It's showing me pretty good conservation of my already artificial habitat type, my greenery. And altogether, so the proper design of the greenery with some parts with these still meadows or lawns or open areas with only grass for playing, the playground for the kids and so on, but it's still some more natural patches inside and a higher diversity of microhabitats will help to find this balance between the conservation role of this of the greeneries and also the recreational role of the greeneries. But when we are talking about urbanization, as I said before, it's not only changing your habitat types or your landscape, but it's also changing the environment. And here, as I said before, we are talking about the urban heat island. This is a study I'm carrying during the last 3 years already, in collaboration with the University of Regensburg in Germany. So the urban heat island, as I said, and here is more clear, is the increase of the temperatures in the middle, in the city center, within the city, because of the reduction of the airflow and also the release of heat from the buildings and from the pavement and the cars and different devices that we have at home, for example, like the air conditioning. And altogether is creating this difference of temperatures between the city center and the surrounding natural habitats. During this study, we wanted to check how or if the higher temperatures in the city is facilitating the foraging of the Lassus niger and during the night. I started that because most of the study, or as far as I know, all the studies that carry out during the, or in the, to check the urbanization are carrying out during the day. Of course, this is much more comfortable than being awake the whole night and counting ants as we did. But if I really want to check this urban heat island effect, I have to do it when the sun is not warming the land, because it's during the night when you go out and you go for a walk, and when you are in the city center, you are still wearing this summer t-shirt because it's warm. But if you have your cottage or your summer house in the natural habitat surroundings, maybe this summer t-shirt is not enough and you have to take a cardigan, for example. So these differences for us is not a big deal, let's say, because we are at home and we have everything. But for us, it will mark the difference between keep foraging, gathering food, and looking for a new place for nesting or different sources, or just stay within the nest and quit or slow down the development of my colony. But in cities, we also have a different things, that is the illumination or the light pollution. And for us, it's very comfortable because you go out, you can work, and everything is nice, it's comfortable, it's not dark and scary. But it might also have an influence on them, because there are some studies showing that with light, the deposition of pheromones is reduced. So maybe if you are saving energy from producing these pheromones, you can allow yourself to keep foraging for longer. And also, we wanted to know whether this higher foraging activity will influence the colony growth because of a higher gathering of food. Here I will show preliminary results because I'm still analyzing the data. We just finished to collect the last data not too long ago, this summer. So we didn't have the time for analyzing everything. And I will show you how we did it. So first, we made our samplings in Poland and in Germany. Why two different places? Well, they are here, they are neighbor countries, but still their temperature is a bit different. And what was much more important for me, the structure of their cities is different. And if we are thinking about the urban heat island structure of the city, will allow or not a higher airflow, a higher refreshment of the ground, and a higher overheating, for example, if it's less green. To carry out this study, we did a field study and a laboratory one. The field study to see the real foraging activity, and the laboratory study to test the different factors, the illumination and the temperature, without any other influences from the surroundings, like different smells, people dropping food, and so on. Here are the places, oh, it moved. Here are the places in Poland that we sampled. One is Drastic in the north, near the sea. The second is Warsaw, where I'm living and working, here in the center of the city, of the country. And the other is Krakow, it's close to the Tatra Mountains. So three cities, but it's still a bit different because of the possible influence of the weather. And here, in Germany, we it also moved a bit. In Germany, we sampled Berlin and Regensburg, a huge city and small city. That's a town with small buildings and quite a lot of green areas here. In all these 5 cities, we sampled urban habitats. So we try to find the typical park that you have here in Europe, like this. Your sidewalk, your grass here, and some tree. And natural habitats at the surroundings. So to check them, we select the city wonders and we move out to the natural habitat. There we place the baits with sugar water and tuna. We place a thermometer at the ground, and this is a small light just to find where you have the bites during the night. Baits were placed randomly, so to avoid that ants are selecting one or another bait because it's close to the nest or not, we were placing them randomly. And the groups of baits were separated among them for 5 meters, and 2 transits of 10 pit foot traps, 10 baits, group of baits were placed in the ground in each habitat type, separated by 10 meters. So we did it twice per place with a difference of 3 days among the samplings. And finally, we also check a part of, sorry, I will be back here because jumped, sorry. A part of this checking of activity, we also checked how ants are intaking the different food type. Again, we were using sugar, but we also here we select tuna and tuna with oil, and all of them in the same way. They were all in a liquid state. So the texture of the food, it wasn't a factor for the selection. And we check also the fat content. For that, we went to the field and we collect the giants and the workers just before the nuptial flight. And because I didn't have any photo of this study, of this part of the study, I put a photo of my students, all of them collecting these giants and workers in the field. And they looked happy, I think. And finally, as I said, we did the laboratory setup to control also the temperature and the light. And how we did it? Well, we collect the colonies, 3 colonies per each habitat type and each park. So 9 colonies from urban habitats and 9 colonies from natural habitats. And we split half of the colonies with larvae, with broods in general, to each one of the different thermochambers. One thermochamber was simulating the urban habitats with higher temperatures and a light dark cycle. The second chamber was simulating the natural habitats with lower temperatures and simulating also the dark light cycle. And the third one was to check the light pollution. So we set up the same low temperatures as in the natural habitats, but we had continuous illumination. So we built very amazing artificial nests, all of them with same number of workers and larvae, place them in the thermochamber, and this is how it looks like in the reality. All these nests were having food and libidum, so proteins and sugar solution. And what we did for not disturbing the ants and interfere in their activity was, the day 1, we were feeding the ants. During the day 2 to the day 4, we made the observations without touching the colonies. Day 5, we fed the ants again, and again observations started 24 hours later, from the day 6 to the day 8. And every half an hour, there was a camera inside. This is a camera connected to a BlackBerry Pi, and it was programmed to take a photo every 30 minutes. After all of these photos, we had 6 days of photos every half an hour from each chamber, and we had to count the ants of all the photos present here in the photogene arena. So the results show that the temperature, or the average temperature within the city, was higher than in the natural habitats, and the number of workers or active workers during the night was increasing accordingly to the increase of the temperatures. When we were comparing the different cities in the in Dansk or in Poland, we see that all of them follow the same pattern, increasing the activity when it's increasing the temperature, but the increase will differ among the different cities. So here we are also having some effects of probably the type of weather that we were having in all cities. And this weather will enhance or not the effect of the urban heat island on the activity. But when we are putting together all that we had in the activity of ants during the night, or the number of active baits during the night, in all the cities, we saw a different pattern between the two countries. So, whereas in Poland, we saw that there is a higher, I don't know why it disappeared, there is a higher number of active baits in the urban habitats, not in Krakow. We saw that in Germany is totally opposite. Is the natural habitats the most active habitat type, or the colonies from natural habitats were the most active ones. So that's something that I really don't have any explanation, and we have to think about whether is the structure of the city whether it's a real effect of the weather, or whether there is something different and is more than one species, because Lassus niger is actually one species that has not been studied for long. And lately, some common species, like, for example, Tapinoma nigerrimum, has shown to be a cryptic group composed for 4 different species. So actually, I do not have an answer for that. Maybe it's a mix of all of that, but it's something that we still have to check. But when we were having the this study, this part, the laboratory was done with only colonies from one side, well, from one city. So here we are avoiding the problem of this possible different species, and of course we are absolutely avoiding the problems or the influence of the habitat, because we are controlling all the factors within the thermal chambers. And the results are showing that, on the contrary of what we expected, natural populations are having a higher response to the increase of the temperature in all chambers, in the high, in the low, and in the illuminated chamber. And also the light, well, this one we expected. The light shows to have some influence on the foraging activity, increasing the foraging activity or helping to forage. So when we are comparing these low chambers with darkness and light, we can see how the activity is higher in this chamber with the continuous illumination. So if we put together maybe everything, all the factors influencing the temperature and the light, maybe in urban habitats, the foraging activity can be enhanced much more than that we see here in our experiment. But what is really surprising is this part. But still, there are some studies showing, carried out in Temnotoras curvispinosus, showing that urban ants are not well adapted to high temperatures. So despite we think that ants are adapting to high temperatures, or maybe it's just a plasticity, it seems that natural populations are much more positively influenced by the high temperatures that urban populations. And this urban population seems to have lost this capacity or this response. But I also think that it might be happening something else. And it's not just a misadaptation, but maybe when you have the chance of being foraging 24 hours per day, why to struggle? So maybe we do not see the increase of temperatures just because you have the chance to be foraging the whole time. And maybe you just keep doing that, because we also know that ants are quite linked to the activity that they are learning. And oh, sorry, I will, it's not. So, and finally, when we were checking the nutrition and the food intake between the two habitat types, also linked with the foraging, because if you are foraging longer, you can intake longer food, amount of food. We saw that checking the fat content of the giants and the workers collected before the nuptial flight. So they have been absolutely and only fed by the workers during the foraging. Giants from natural habitats had a higher amount of fat than the giants from urban habitats. So it seems that the giants from natural habitats are feeding much more than those urban giants, but no differences were found in the type or in the case of the workers. But one more time, because all researchers are showing you something, a new surprise to keep checking that. If I'm having a higher amount of fat, maybe I'm consuming or is linked to a higher consumption of fat content. And in natural habitat, it's supposed to be that we have a higher number of arthropods, so a higher number of fat source. But when we are checking the food intake, and sorry, I forgot to say that usually, well, there is something that we don't know. Ants are collecting what they like, what they need, or what they are used to. So here we find again the contradiction. When we are checking, when we were checking the amount of food that they are taking, or what they were selecting, ants in general were selecting a higher number of or a higher amount of sugar, which is absolutely linked with the ecology. But when we were comparing the amount of fat and the amount of sugar or carbohydrates that ants were taking, the natural populations were collecting a lower amount of fatty food than the urban ants. They were also collecting lower amount of fatty food when they had the choice between fatty proteins and just proteins. The spike is not significant, and they were intaking more sugar. So those same ants that are higher fat content were the ones collecting less fatty food. So again, I do not have any explanation for that, and we really need to analyze that and find out what's the problem. It's just urban environments are also struggling or are also endangering the diet of the ants, or they, like kids now, taking much more sugar or much more fat, or how it is. So the conclusions show that urban and natural populations show a different response to the increase of the temperatures. Oh. Urban environments leads to changes in ants, but these changes are linked to the increase of the temperatures in a different way depending on the different cities, or the different structure, or the different environmental characteristics, like the weather. We don't know. We also know that the artificial illumination during nights have an effect on ants, seems to have an effect on ants, and can enhance this activity, also facilitated by the temperatures. And finally, we need much more further and deeper studies to really find out the solution for all of these doubts that have arisen with this study. And finally, I would like to thank everybody that have been helping me, have been working hard, have analyzed videos and sampling, and teach me a lot. And they are my students, Dani, Michał, Paulina. Paulina was working a lot and she's here now. Volga and Sophie, also a great worker and who collaborate a lot with that. My collaborators. Oh, sorry, I lost my voice. Sorry. My collaborators, Thomas Drastic, it's very difficult for me, his name, and Professor Jürgen Heinze, my professor from the University of Córdoba, who taught me a lot about ants, Joaquín Reyes López, and my colleagues at work, Magdalena Vitek, Luca Pietro Casacci, and Isma Mack. And of course, all the people funding this, all these, the institutions funding these studies, and the city halls helping me to and allowing me to be sampling during nights in their parks. Thank you very much.
Awesome, thank you so much. I know I don't know very much about ants, but I feel like I have a better understanding about how they're functioning, at least in the city. I think something that struck me the most when you're talking is about how difficult it is to do these ecological studies, to really try to separate all the different effects. And so it's cool that you have the laboratory study to at least start to disentangle that, but you know, it's difficult, but that's also how the systems are working. They're very complex systems. So
yeah. Because as you said, everything is connected. You cannot separate a thing from another. And so if you're studying just a factor, your ants or your animals or your plants are not isolated. They also have the effect of other things. Like if you are smelling the perfume, you are smelling it better if you have some wind, because it's coming stronger to you. So this this phrase is sample is like showing how everything is working. In nature, some things, or like the activity, can be damped if you have maybe a higher humidity and with a bit of wind. So the soil surface is getting cooler faster, and your activity will decrease, because ants' activity is fully linked to the temperature. Or for example, let's say sometimes we were placing the baits, and ants are moving around and they are ignoring the bait. As I said, ants are also learning something. If there is some amateur here, you know that when you are placing the dish, your nest, and then you place the food, ants learn. And like 1 week later, you have your nest, and you have a layer of sand or soil, and you arrive and you place the bait. You do not have to put the food yet when you already have your ants coming up. They are learning. And for example, if you are placing your baits and there is a food source that they are really fond before you were placing the baits, they are also able or capable to feel the quality of the bait. There is Tommy, and my collaborator from Regensburg. He's working with that. He has some talks about that. He's what he's doing is showing different food source with different quality, and they learn the good quality. So when he's putting lower quality, they don't like it. And if you are having low quality and then you are putting higher quality, they go to this food. So these factors are affecting You cannot control what you had before in the nature. You cannot control what you have at the moment. You cannot control that it's going to rain, or maybe it was rainy before. So in that case, you also need the laboratory studies to control as much as possible, but you will never get to control everything, because ants also have a sort of personality. And you have more aggressive ones, you have more lazy ones, you have colonies that are very active, and you have colonies very inactive. So even though you want to find the better setup, you will never have it. You will always have this factor saying you, hi, I'm here. So yeah, it's very important to try to control everything. And some people think that only laboratory experiments are like reliable. Some people is saying that not field laboratory are reliable. In my opinion, I think that you have to combine both, because if you only have the laboratory experiments, you do not have the reality, because nature is not like that, and you will never have everything controlled. But if you have nature, maybe you do not really get to understand the real influence of this factor. So in my case, I would say that I'm very lucky, because in my teamwork, but also the people I know, I have a mix of people from strict laboratory and strict field studies, and I really have a lot of help from them when I'm having some doubts, when we are discussing something. And really, when you put it all together, it really you can really get to find something or a good setup, but never perfect. There is a picture, there was a drawing that I received months ago, that there was it's just a guy thinking about a study, scientific study, super happy, wow. Then he's performing the study and is even happier. He's analyzing the data, so excited, and finally he's super sad, like results. Then I don't know, maybe some people is super lucky and always fit what they expect with what they have. But in my case, I always have some surprise, which is also nice, because you keep searching for that. Like now, we did that, and now we have lots of questions to keep going and to try to understand what's going on.
Right, I guess the best or worst kind of research you'll end up with more, right, 10 more questions of other things that you want to find out. We do have a question in the chat, and if anybody here wants to either type your question if you have one, or if you want to ask, feel free. We had a question from Jack earlier. Have you done any sequencing on the ants between the two countries, or I would say at all, to see if incipient speciation is taking place?
There is my collaborators from Regensburg, they were starting to do the sequencing, but only to check whether there is some differences between the different populations there. For now, like the money of the project is not allowing me to make any sequencing, and you know, that's how it works. You sometimes have the budget and sometimes not. But in my opinion, I don't know, or I'm not that sure if there is some speciation suddenly. Maybe they are different, but it's still like we were very careful, like selecting the places to avoid things like, for example, avoiding putting places that can lead you to take or to collect glasses platithorax instead of Niger, or like a background that can lead you to create some lassus tabularum instead of the Niger. So we did our best in this habitat selection to reduce as much as possible the differences or to have different species, but maybe there are some, as I said, it's happening with some different groups like this Tapinoma nigerimum group now. Before, for many years, only Tapinoma nigerimum species, or I don't really know. And probably we will have at some point some sequencing. Actually, I'm planning to do some sequencing and in the future for different aspects. But right now, if I'm lucky, because I'm applying for a grant in December and I get it, it will be possible. If not, we will have to wait for next next opportunity. But yeah, it would be nice and it would be interesting, of course, because we all have this doubt. It's different species, it's just habitat structure, it's weather, it's all together.
Also, I was thinking about another question about the pitfall traps. So I guess this is one of the most common ways in insect research to collect, but you mentioned that there's some kind of course, you only will get the things that happen to fall in. So is this the best method, or are there other kinds of methods that could be used in a different situation, or that are very situational?
It's a bit difficult to answer. I will tell you why. Because depending on the habitat type and the species that you will have a larger or smaller number of species that will forage at the surface. So if you have a habitat type with a high number of epigenic species, I would say that's the perfect one. Because even if you want to sample the whole habitat type, every centimeter, you might not find all species, because you can have a very small nest in small piece of wood, or they are eventually super deep in the ground, or many different things. And if you are not at the moment that they are foraging, because not all the ants are foraging at the same time, there is this different activity foraging along the day. You have those foraging in the morning, and it's super hot. You have all the other species, so there is a substitution of a species along the day. So maybe you won't find this species. There are some others that never come out, like the Leptanilla or some Solenopsis. They not always come out. Of course, we all know the Invicta is so noisy, but there are some other a lot of species of Solenopsis. Or you have also the parasites. So some are like well, Strongylonatus is coming out, but Anergatus, for example, is not coming out. It's never coming out. So it's very difficult that you will get everything. But when you have a methodology working for 48 hours continuously, or 72 hours, you are increasing the possibility of capturing different species. But there is also the bad part of that maybe it's not needed always to kill such amount of ants, because many times you are really killing a lot of ants, and we don't really need that. So I would say that maybe punctually for some study in the habitat type, maybe we can place the pitfall traps and have a general idea of what we have. But if we do not need it, if we are taking some specific factor or some specific thing in the habitat, why to kill that? We can just choose one ant, the most specific one for that is feeding or fulfilling our requirements without the need of using all the others. So like, is the best or not? Depends of the species, depends of what you want.
And we saw, I think the picture that we used when we were advertising was the one that you have the tube to the I don't know what they call it, the ant sucker.
There are many different names. So you have the exhaustor, the whatever, we call it butter. Or actually, with our pronunciation, I have to say we call it putor. But it's not the right pronunciation. But if you come to my department, everybody's asking for the putor. And actually, you can build it. You do not need to have anything special. Actually, this one, I built it. So you just have some glass. Okay, I will show you something very quickly. And here I have. Look, actually, we are usually building it because what you buy is very nice when you have to collect 1 or 2 ants. But for example, to collect a large amount of Lassus niger, because I'm not collecting the nest. I don't need the queen, so I'm not collecting the nest. There is no need to kill the colony. So you have a plastic glass. I don't have it now, I have this there, but let's imagine that this is a plastic glass, like for like this, let's say, hearing analysis. So you have the lid, you make a hole here and a hole here. And then you buy a pipe. Look, this one is from one, what you buy from the different entomological shops. So you buy 2 pipes. In one of them, you put a piece here, a piece of cloth. Okay, like a net. And the other is free. Okay, so let's say you have put one tube here and another nearby. And now you will have something like this. So from the pipe where you put the clothes, you just suck. And from the other, you are collecting the ants and you are dropping here. It's
so the cloth will help to,
yeah, otherwise you will need the
filter.
ant, the sand, and everything.
Very cool.
So when you are just collecting cement, those from the entomological shops are amazing. I have them. They are small, they are very useful and comfortable. But if you are collecting big amounts for our colony for these experiments, then we are building them with a larger jars, so you do not have to pour them and close again and so on.
Or that's even something I wonder if, you were to do something with high school students or something, like you could have like a DIY version to collect.
Well, they are usually in the Institute of Zoology there in Warsaw. We have students, they are coming from the university, but sometimes they are also coming from the high school. Sometimes it's the colleague of someone that who would like to know about ants, and they are coming. So they are actually learning and asking. And when they are coming so young with any, let's say, they do not need to do the master thesis or the bachelor thesis, they are just coming. It's very nice because they really are enjoying that. They are asking, they are very enthusiastic, and they really are curious about everything. And I'm a very bad teacher, I have to say. I don't like to explain, and well, I hope that the presentation was more or less okay. But I like when I have some students, 1, 2, 3, I don't care, and they are asking. So it's not like this pattern that you are just blah, blah, blah. No, you are just showing. You bring them outside, you go to the garden, you go to the nature, you take one hand and the other. And this is different. This is like a conversation. And this one, I like it.
Yeah, definitely. It's easier to explain when you have them and things in front of you than to just.
Well, I think I would say that it's easier to explain when you have people with interest. You know, when you go to lessons, I have been a student and I wasn't interested in all the lessons I had. Some of them, unfortunately, I had to pass the exam to be a biologist. And some others, I was fascinated. And that's the same, because we are all different and we have different curiosities. So when you're having these kids, that they don't have to be there, but they are because they want to. This is very nice because you can teach them. Unfortunately, we never have time enough. Like you are always running from one experiment to another for analyzing data. Now you have a conference. Now you have to apply for a grant, and now you have to run again. And this part is not maybe allowing you to spend the time that you would like to spend with them. But what can we do? We also have to survive somehow.
Finding the balance, you know, that's good though. That's great because right, even if they also just to have the exposure, even if they come and they say, oh, actually, I don't really like this, that's good because then they've tried it out and they know. So I think that's important. Cool. Does anyone else have any other questions or comments you'd like to make before we finish it up?
I guess I just have a quick question just about ant identification. What are some like common anatomy features on an ant that you use to differentiate species? I saw some coloration differences, size differences, but I'm curious if there's anything that's commonly different between species.
Well, I wouldn't take coloration as a very good factor or a very good character to identify because colors are changing. Like, for example, Mesor barbarus in South Spain has a really red head. And if you're moving to North Spain, the head is darker. But it's not a different species, it's just the temperature that the nest is having during the development of the larvae. Also, for example, Myrmica species are super light when they are born and they are getting darker with age. So if you have a very light one and a very dark one, maybe it's the same species, just different age. So coloration is not the best character that you have. I like to, when I'm identifying, or I was sort of, my professors sort of taught me like that, and I like this methodology, or maybe he was teaching me in similar way and then I modified to my own way. But I do not like to teach identification to the students with this identification key, and then you have the ant and you follow all the characters. I don't like it because you memorize different numbers and names and different things in 1 day, 2 days, and in 2 weeks you will forget it. I put them a bunch of ants, different species, and I tell them, split them into different groups that you think, like a group of all similar ones, and split the groups of different ones. And then why they are different. Tell me how they are different and what. And of course, they are not always telling you right, but this is not a trick. It's just they are, for me, it's very important that they are making their own picture of the ants. And once you reach to see the difference on your own, you get this picture in your head. And now we jump to the key. And now we start putting names. Now we learn what is the seta, what is the scapus, what is the tibia and the antennal suckers and things like that. But I want you to have the picture, because this picture is yours and you will always remember this picture. For me, I always tell it, the cataglyphis is a model, like long antennas, long legs, and very slim, you know, super nice. This is a, you know, like a model. And then I have the tank. And so like the mimica for me are like tank. Very good. So this is the picture, the general picture I have. And when I go to this to the field, I see it and I see the tank and the other. And then, of course, to reach the species, you need to follow the characters. But when they are learning one petiole and post petiole, and whatever, and down, and the 11 antennal segments or 12 antennal segments, and then you ask, what is it? What is the temnothorax? And they say you, it's a small ant with a petiole and post petiole and 11 antennal segments, and blah, 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 blah. And then you put the temnothorax and you put the myrmica nearby, and they don't know what is it. What for? So for me, it's important to put this, make the picture, and we start putting names. Because you won't forget the pictures and names you will always find in the key. And actually, when you will read the key 200 times, you won't forget the names as well.
That makes a lot of sense to me. That's similar to plant ID, I think, a lot. Oh,
yeah. We're gonna have to go and see if we can try to collect some ants and see what we have. Because we have the botanical garden, so that might be a really good you know, high diversity here, because we have all the different habitat types and everything like this.
Yeah.
Cool. Okay, well, thank you so much for coming and speaking with us and for staying up late to entertain our, you know, questions and everything. Thanks everybody for coming. We'll have another talk coming up at the very end of the month. And hopefully we'll have some walks and everything in between them. So hopefully we'll see you guys soon. And have a great rest of your evening.
Thank you, everybody.